So we're going to start on our next topic, which is metabolism. And we've already talked a little bit about metabolism in the sense that we've talked about the idea that one of the characteristics of living organisms is they all have metabolism. And metabolism is really just chemical reactions. It happens to be chemical reactions that happen either inside cells or because of something that cells do. Sometimes the chemical reaction actually takes place outside of cells. When we look at metabolism, keep in mind what we've said before. A typical cell has about 10,000 proteins in it. And most of those proteins are enzymes. And remember that enzymes control metabolic reactions, chemical reactions. So this means that a typical cell can do about 10,000 different reactions. Some cells can do more, some cells can do less, but yeah, 10,000 is a nice round number, so we'll stick with it. But when we look at these reactions, what we want to keep in mind is they don't really happen by themselves. They're connected. Just like if you were giving somebody instructions how to get to your house and you weren't just going to say, go to Google's Maps. If you were going to actually give them instructions, you would say, well, go down the Bishop Ford Expressway, get off on the exit at 162nd Street. But they would get off on the 162nd Street exit after they went down the Bishop Ford Expressway. So they would have to do things in a certain order. And that's what we're going to see about metabolism. We're going to have a lot of the chemical reactions that take place in cells, that reaction A has to happen before reaction B can happen. So it's not just as simple as everything happens haphazardly when it gets in the mood. So to think about metabolism, we want to keep in mind something we said when we talked about chemistry. Everything in the universe is made up of only two things: matter, and matter has mass and takes up space, or has volume if you want to be more technical, and energy. And energy is defined as the ability to do works. Now, work is defined the way it's defined in physics. So we won't worry about the technical definition, but when you go to work, if you work at a job and you're moving boxes from one shelf to another, that is work the way it's defined in physics. But work the way it's defined in physics is actually brain cells communicating with each other, thinking, in other words. So there are a lot of different types of work. So when we think of the ability to do work, we don't want to think of that in terms of only going to a job. Now, one of the things we said was that you can't create or destroy energy, just like you can't create or destroy matter. And the laws that govern energy are described in the laws of thermodynamics. Dynamics refers to things that are changing. Thermo really refers to heat, but in this sense, it's used in a more general sense to just mean any form of energy. Heat is one form of energy. So the first law of thermodynamics is what we talked about when we discussed chemistry. Energy can't be created and it can't be destroyed. The energy that we have in the reactants has to be the same amount of energy that we have in the products. So relatively simple. Now remember, we said when we look at a chemical equation, we see energy in two different forms. One of those forms is hard to see because we don't see it written. But whenever we have a molecule, whenever we have atoms bonded together, there's energy in chemical bonds. Remember, we just said in the last section, in diffusion, atoms, molecules, ions are constantly vibrating, they're constantly moving around. And this means if we want to anchor one atom to another to make a molecule, we have to restrict their motion. So this takes some energy. So there's energy in chemical bonds. And there's also sometimes free energy, which is energy that isn't in chemical bonds. So if we look at an equation, one that we're going to become very familiar with over the next week or so, We have a molecule of glucose. Notice we have one molecule. We know it's one because there's no number out in front of the glucose. We're going to combine that with 6 molecules of oxygen. We're going to make as products 6 molecules of carbon dioxide, 6 molecules of water, and energy. This is free energy. It's not contained in chemical bonds. That's why it's written separately. If it was in chemical bonds, we wouldn't see it. So in this case, this is the energy in the products. So this means that if we look at this, and in fact, you could do this reaction, if you took something like table sugar and you put it in a spoon, and you put a little lump of table sugar on that spoon, and then you lit it with a match, it would burn. And what you would make from that would be carbon dioxide and water, and energy would be the light that you could see, the flame that it would give off when it was burning, and also the heat. Wouldn't exactly be like a roaring campfire, but there would be a little bit of heat given off as well. So all we can do is change energy from one form to another. So in this particular equation, we have some energy in the chemical bonds of glucose. We have some energy in the chemical bonds of O2, oxygen gas. At the end, we've moved some of that energy into chemical bonds in carbon dioxide, and some of it into chemical bonds in water. And some of it has kind of come loose, so to speak. It's not in chemical bonds anymore. So we can change energy. We can change which chemical bond the energy is found in. We can't create it. We can't destroy it. So we want to keep that in mind. Now, there are a lot of different forms of energy. Light is one form of energy, heat, mechanical energy, which is sometimes referred to as energy of movement, chemical energy, the energy that's in the bonds. These are some of the main ones that we'll come across in biology, but there are other forms as well. Now, all the different types of energy can be subdivided into just 2 categories. When we look at energy, energy is either potential energy or it's kinetic energy. Potential energy is energy that could do work at some point. So the energy in chemical bonds is a good example of potential energy. If you have some gasoline in your car engine, or if you have some charcoal briquettes in your grill, they could release energy. But right now, all the energy in those charcoal briquettes or in that gasoline in your car engine is just in the chemical bonds. That energy will be released if you break those bonds, like we saw up here. The other type of energy is kinetic energy. Kinetic energy is technically defined as energy that an object has as a result of moving. So if you think about 2 cars that collide with each other, the reason there's some damage is because those cars have some kinetic energy when they're moving. And if they collide, they transfer some of that energy to the other car, which may cause pieces of that other car to bend or break. Or if you ever saw those old Roadrunner cartoons, when Wile E. Coyote falls down the cliff and he lands on the ground, he always like makes an indentation, digs a little outline of his body, a couple inches into the ground. That's because he has kinetic energy. And when he hits the ground, that kinetic energy gets transferred to the ground itself. So kinetic energy is defined as energy of movement, but another way to think of it is it's energy that is doing work right now. So it doesn't just have the potential to do work, it is doing work. The second law of thermodynamics says the amount of entropy in a closed system will increase over time. What does that mean? Well, practical way to think about this is something that we mentioned before. When energy changes from one form to another, the change is never 100% efficient. So if you think about burning gasoline in your car engine, you would like to convert the energy in the chemical bonds of the gasoline into mechanical energy that moves your car. That's how you're going to get someplace. The problem is this conversion isn't 100% efficient. So if you've ever had car problems and after a while you've had to get out of the car and open the hood of the engine, you probably know, if you didn't know the first time, you certainly learned the hard way, you probably know that you don't want to put your hands on the car engine. Why? Because it gets hot when the motor is running. Why? Because some of the energy from the chemical bonds in the gasoline gets converted to kinetic energy that moves your car, but some of the energy gets converted into heat energy. So the engine block gets hot. So energy conversions are never 100% efficient. So we want to keep that in mind. And usually when we talk about energy isn't completely changed to an alternate form, heat energy is usually the least useful form of energy. So usually heat energy is what we're talking about with that free energy. Heat energy is kind of, in a sense, the lowest form of energy. There aren't very many systems that can actually use this, we'll call it waste heat. We can use it because we're what some people call warm-blooded animals. When our metabolic reactions lose some heat, we can basically hold on to this heat, and then we use it to maintain our body temperature. And if you think about our body temperature, human body temperature is 98.6 degrees Fahrenheit. Yours might be a little bit higher, a little bit lower. There is individual variation, but just think about that. If your house was 98.6 degrees Fahrenheit every day, or if you lived in an environment where it was always 98.6 degrees outside, that would be incredibly hot. So because of the fact that our bodies are using heat to maintain their body temperature, our bodies work as a furnace, much better than as an air conditioner. Think about if we had some really nice weather and our high temperature tomorrow was 70 degrees. You could probably go outside and be perfectly comfortable. But think about 70 degrees. That's 28 degrees below human body temperature. If you went outside tomorrow and it was 126 degrees, if it was 28 degrees above your body temperature, you would not be able to get comfortable no matter what you did. Because our body works much better as a furnace. We cope much better with cold temperatures than we do with warm temperatures. So we want to keep that in mind. Now, when we look at metabolic reactions, there's a lot of different ways that we can think of them. One way we can think about them is what happens to the amount of energy in the chemical bonds. So let's think first about something called an endergonic, or sometimes an endothermic reaction. So think of endo. Endo means in. Gonic and thermic refer to the energy. I know gonic is not something you're familiar with, but it's a technical chemical term. In these types of reactions, energy goes in. So this means that energy goes into the reactants. So the products have more energy in their chemical bonds than the reactants started out with. And this always has the basic format of something like this. The reactants, in this case, our reactants are carbon dioxide and water. The reactants plus some energy gives us the products. Now, the fact that we write energy on the reactant side of the equation and we don't see energy on the product side, this means that this energy, however much energy it was, is stored in the chemical bonds of glucose and the chemical bonds of oxygen. So the chemical bonds of our products have more energy than the chemical bonds of the reactants. Why? Because they absorb some energy. Now, this is actually the equation for a metabolic process that we'll look at called photosynthesis. So the energy for this comes from sunlight. And the energy is stored mainly in the glucose. There's not very much stuff stored in the oxygen. A little bit, but not much. So when we look at metabolic reactions, a lot of times the energy that we need for a reaction that's going to absorb energy is going to come from another reaction that released energy. So reactions are kind of combined. Something that releases energy occurs, then that energy it releases is used by an energy reaction that needs to absorb energy. Now, if we're thinking about the energy in the chemical bonds, the other type of reaction we see is something called an exergonic or an exothermic reaction. So think of exit. Exit is where you go out. So in an exergonic or an exothermic reaction, the reactants lose some energy. The energy goes out of the reactants. So the reactants contain more energy in their chemical bonds than the products do. So we always see something in this general format. The reactants make some products and the free energy is written on the product side of the equation. So this represents some energy that's lost. So in this equation, and I mentioned this before, we're combining glucose with oxygen, and the energy that we give off if you lit a match, this would be the heat and the light that you saw from the fire you would make. Now this reaction also happens in your cells, but it happens very slowly in a large number of steps. And because it happens very slowly in a large number of steps, we don't really have an open fire going on in your cells. So we'll talk about that later. So let's think about looking at this graphically. So let's think about an endergonic reaction. And on our y-axis, we're going to graph the amount of energy in the chemical bonds. So notice we're not necessarily graphing the free energy on this. And the x-axis just represents our reaction. So how long it takes in terms of time. And notice for metabolic reactions, we're talking about a fraction of a second. So our reactants are over here on the left, and by the time we get done over here on the right, we have our products. So in an endergonic reaction, what we have is the reactants start out with a certain amount of energy in their bonds. That's where this black line is down at the bottom. And the products end up with a certain amount of energy in their chemical bonds. That's where this flat line is at the end. Now notice, since this type of reaction absorbs energy, the products have to be at a higher level than the reactants started at. But notice we don't just go straight from the reactants to the products. We have to go over this little hill here, this kind of little hump. And this little hump, the height of this hump, so we're going to measure that height. We're going to start down here with how much energy is in the chemical bonds of the reactants. We're going to go up to the top of that height. That's called the activation energy. That's how much energy this reaction needs just to get started. So we'll look at some examples of this in a moment. Now, how much energy was actually absorbed in this reaction? How much would the free energy that's over on the reactant side of the equation be? Well, if we measured it, that would be the difference in height between where the reactant started out and where the products ended up. That would be how much energy is absorbed from that free energy. Now, an exergonic reaction, in a sense, is the reverse. Again, our reactants are over here on the left, our products are on the right. The reactants start out with this much energy in their chemical bonds. The products end up down here. So the reactants have lost some energy. So notice again the difference in height between where the reactants start and where the products end up. This represents the free energy in our equation, but this time This is the free energy in the products. This is how much energy is being given off, how much light and heat burning that glucose generates. And notice again, we don't go straight from the reactants to the products. We have to get over a little bit of a hump. Think about the example that I've been using for you, that you could put some glucose in a spoon and you could light that glucose on fire and it would give off some heat and it would give off some light. That would be the free energy that we see here. But think about how you're going to start this glucose burning. What did I say? You're going to light it with a match. That match is the activation energy. That's what we have to kind of use to start this reaction happening. So we've always got some activation energy, even if the products end up with less energy than the reactants. So we want to keep that in mind. Now let's think about what we have to have a reaction actually occur. Keep in mind there are, 10,000, 11,000 chemicals in a typical cell. Some of the chemicals like water, there's a lot of water molecules. Other chemicals, there's only a few. So, one idea to think about all the different molecules that are in a cell. If you think about the room that you're in, imagine filling that room up with grains of sand. Every grain of sand would represent a different molecule inside that room or inside that cell, if we want to think of that whole room as being one cell. And there would be about 10 or 11,000 different colors of grains of sand. And each color would represent one different kind of chemical. About half of the grains of sand would be water. Everything else, there would be some. There'd be a lot of glucose, but not 50%. There'd be a lot of ATP, but not 50%. So we've got all these different molecules in the cell. So what do we have to have if we're going to look at a reaction where we have two chemicals that are going to collide? One of the chemicals is a molecule of nitrogen and connected to some oxygen atoms. We can see O's written on the inside. Excuse me. One is a carbon atom connected to some oxygens. So first thing that has to happen is the correct particles have to collide. So if we're looking at this chemical reaction, we have to have the right two things collide. Remember what I just said, there's 10,000 molecules in our cell, 10,000 different kinds of molecules. So if the wrong two molecules collide, nothing happens. If we look at the reactants, they have to be oriented in the correct direction. In this case, this oxygen actually has to be facing the nitrogen. If it's facing away from the nitrogen, nothing happens. When the two reactants collide, they have to collide fast enough. If they don't collide fast enough, they just kind of bounce off each other, kind of like two balls if you're playing pool. And if the right two particles collide, if they collide facing the correct direction, and if they have enough energy when they collide, and energy is really just measured by speed or temperature, it's kind of the same thing at a molecular level. If they have enough energy, they've got to have enough to break the old bonds and make the new ones. Then we get a reaction. So notice in this third case, we get a reaction at the end. Nitrogen only has 2 O's bonded to it. The carbon has 2. Carbon started out with 1, nitrogen started out with 3. So we've got to have the correct particles collide. They have to collide at the correct orientation, and they have to have enough energy to break the old bonds and make new bonds. So when I think of particles colliding, I always think of, at least where I went to school, like one of the first dances that they held like in junior high or at the end of grade school. So you have to have the right particles collide. A guy and a girl have to face each other. They actually have to face each other. So they have to have the correct orientation. And they have to have enough energy to react. Somebody has to actually ask the other person to react. So you've got to have all these things in place. If one or more of these is missing, nothing happens. Now, how can you make a chemical reaction more likely to occur? Well, one of the easiest ways is to increase the temperature. If temperature goes up about 10 degrees Celsius, which is about 18 degrees Fahrenheit, this doubles the rate of metabolic reactions in a cell. If the temperature goes down 10 degrees Celsius or 18 degrees Fahrenheit, the rate of the chemical reactions is only half of what it was. So this is important in things like hibernation. When a bear hibernates in the winter, its body temperature drops 18 degrees. So now its metabolism is only half of what it was. It drops another 18 degrees. So now its metabolism is half of half. It's a fourth of what it was. It drops another 18 degrees. So notice now we're talking about 54 degrees Fahrenheit. So now its metabolism is a half times a half times a half. Its metabolism is only an eighth of what it was before. So one of the ways that bears survive hibernation is they eat a lot before they hibernate, so they store a lot of fat, and then their metabolism slows down a lot so that they don't use up their energy stores very quickly. But actually some animals, when they hibernate, if they haven't stored enough food during the summer and fall, they starve to death over the winter. They use up all their energy and their body kind of digests itself and they never wake up. So increasing the temperature can work. We can also increase the concentration of the reactants. You know, if we go back to what we said up here, we said, you know, we're going to only have, whoops, I'll find it. We've got about 10,000 chemicals in here. Well, if we have 20 molecules that can collide, this reaction will go a lot faster if instead of 20, we have 200 molecules of these. Why? Because the more molecules we have, the more likely they are to collide with the correct partner. So increasing the concentration can work. And these two factors are really important in industry, but there's a problem. You can't really increase the temperature in living cells very well. And it might seem like it wouldn't matter, but you can't increase the concentrations of different chemicals. You can adjust these a small amount in living cells, but you really can't adjust them very significantly. So this means that increasing the temperature and increasing the concentrations are not the best way to necessarily solve these issues when we actually look at a living cell. What works better is something like a catalyst. Catalysts speed up chemical reactions, and they're used in a lot of industrial processes. Now, we're going to focus on enzymes. Enzymes are biological catalysts. And almost every metabolic reaction, there's only a half dozen examples that I can think of out of thousands. Almost every metabolic reaction is catalyzed by an enzyme. Now, enzymes are proteins. So remember what we've been talking about. Proteins always work by their three-dimensional shape, which depends on the order of the amino acids. Enzymes are very picky because they only work by their three-dimensional shape. So most enzymes only work with one reactant or a few reactants that have the same shape. The reactant for an enzyme is sometimes called the substrate. So when we say substrate, just think reactant. And as I say, most enzymes are very specific. So they only catalyze one reaction. So they take one reactant, they turn it into one product, and that's all they do. So they're an extreme example of a specialist. Now, enzymes aren't used up in a reaction, so the cell can use them over and over and over again. Now, a minute ago, I compared, a chemical reaction to a dance, especially one of those early dances where everybody's kind of standing by the sides of the wall in the gym. And I always think of enzymes as being like the social studies teacher at my junior high. Miss Etheridge would walk around the gym during the dance, and she would grab a guy and a girl and kind of pull them together. So remember, we said the first thing was we have to have the correct particles collide. So she would get a guy and a girl together, and she would kind of force you to face each other and talk to each other. So you would be at the correct orientation. And she would kind of push you out onto the dance floor. So she kind of gave you the energy, or at least reduced the energy, because then you had to dance a dance or she was going to chase you down again. So when we think about enzymes, enzymes make reactions happen faster. So what's going to happen is the substrate binds to the enzyme. So here we have an enzyme. It has two substrates, this kind of golden molecule, this blue one. These little stars where they attach are called the active site. So notice when the substrate attaches, you can see with the gold one here, and now that the blue one's on, you can see over here. But notice when the substrate attaches to the enzyme, it changes its shape. And the shape change is called the induced fit model. Now, notice that when the enzyme grabs onto the substrates, it positions them in a way that is the correct orientation. In this case, kind of the right arm of our golden substrate has to be next to the left arm of our blue substrate. So the substrate grabs these. Once it grabs them, we have something called the enzyme substrate complex, or a lot of times it's just called the ES complex, or even just the ES. When the enzyme gets them together, the enzyme kind of stresses the old bonds that are going to get broken so that it's easier. It doesn't take as much activation energy for these two to react. And then once the product is created, in this case, we're joining these two together. Once the product is created, it's released. And now the enzyme can grab two more reactant substrates, put them together, because remember what we said a moment ago, the enzyme is not used up in this process. Keep in mind, there are some enzymes that only pick up one substrate and then they break that substrate down into smaller pieces. So you don't want to think that they always combine things. That's just this particular example. Now, because the enzyme does a lot of the work, because it gets the correct reactants together, because it gets the reactants facing the correct orientation, because it stresses some of the old bonds, so it makes it easier to make new bonds, the enzyme lowers this activation energy. So this hill is going to be a little bit shorter than it is. It's not going to be quite as high. So let's look at a graph showing the activation energy with an enzyme and without an enzyme. So here's that same type of graph. So on our x-axis, we have the reaction. So reactants on the left again, products on the right. And we have the potential energy and the chemical bonds on the y-axis. So higher is more energy, lower is less energy. Notice this is an exergonic or exothermic reaction because the reactants are up here for their amount of energy. The products are down here. So this red line represents the graph of the energy without an enzyme. So the height of this hill from where the reactants start to the top of the activation energy, that's height of that hill, that's the activation energy. So that's without an enzyme. The green line represents what happens with an enzyme. Now notice when we have an enzyme, the reactants start out at the same level. The products end up at the same level. So an enzyme doesn't change the energy in the chemical bonds of the reactants to start with. The enzyme doesn't alter the energy in the chemical bonds of the products at the end. All it does is it makes this hill, this energy of activation, lower with an enzyme. So think of this like climbing a hill. It's a lot easier for molecules to climb this little hill, which means the reaction will occur, than it is for molecules to climb this big red hill. So it takes more energy to climb this big red hill, so these reactions don't happen as often. A lot of times the reactants collide, they have energy, but not enough energy to climb the hill. So the smaller this hill is, the faster the reaction happens, because the more likely it is that the reactants have enough energy. Now, enzymes don't usually work by themselves. They work with other molecules that we call cofactors. Some of the cofactors are coenzymes, and coenzymes aren't proteins, but they are organic molecules. So when we say organic, we know that that means what? They're based on carbon, right? And we will look at some of these. We'll look at a coenzyme called NAD, and NAD picks up hydrogens with a positive charge. And when we look at hydrogen, a lot of times hydrogen is lost in a chemical reaction. When it's lost, NAD or something else has to pick it up. We can't just have loose hydrogen floating around. Notice the N comes from the name of one of the vitamins, niacin. You might remember when we talked about vitamins, when we were talking about different groups of chemicals, we said a lot of them worked as coenzymes. There's another coenzyme called coenzyme A. And coenzyme A transfers something called acyl groups. So usually things that end in YL have this acyl functional group. If you recall, when we talked about chemistry, we said there are, several dozen functional groups. We only focused on a few of the most common ones. Now, one big difference between enzymes and coenzymes, if we look at enzymes, most enzymes only work in one metabolic reaction. Most coenzymes work in lots and lots of different metabolic reactions. They always do the same thing. So, for instance, coenzyme A picks up an acyl group from one molecule that's losing it and transfers it to a different molecule someplace else in the cell. So when we look at NAD, NAD is always going to pick up hydrogen and electrons. So they always do the same thing over and over and over again. Most of the time when we talk about cofactors, we're not talking about the coenzymes. We're just talking about mineral ions. So remember, ions have either a positive or a negative charge. So one enzyme is something called pyruvate kinase, and it needs potassium ions, K+, and magnesium ions, Mg2+. One coenzyme that's used by a lot of enzymes, that works with many enzymes, is calcium ions. And this turns out to be very important for human health. You normally have some calcium ions in your blood. But if your blood levels are too low, like they could be during pregnancy, then what happens is your body will remove calcium from your bones. And it doesn't necessarily have an immediate effect, but this can make a woman more likely to get osteoporosis later in life. One reason that women are more likely than men to get osteoporosis is men don't get pregnant, so they don't lose calcium this way. There are some other ways that you can lose calcium from your bones that can happen to men or women, but this one only happens with women. Now, if we change anything in the chemical environment of an enzyme, it affects the enzyme's activity. So we've already mentioned a few of these. Temperature can change this. The concentration of any substrates can change this. The pH, remember that's how many hydrogen ions there are, H plus, or OH minus ions there are. And so for each of these factors, when we look at enzymes, every enzyme has an optimum. And the optimum is where it works best. And this means that the reaction that it's in charge of will go the fastest at its optimum. So if you look at a lot of enzymes in the human body, their optimum temperature is around 37 degrees Celsius or 98.6 degrees Fahrenheit. Why? Because this is human body temperature. So if you think about it, it makes sense that they would work best at this temperature. On the other hand, if you would look at enzymes from a plant, and there are some reactions in a plant that are basically the same reaction as metabolic reactions that happen in your cells. But if you would look at the enzymes in a plant, plants don't have a body temperature of 37 degrees Celsius. Most plants have a body temperature that is closer to 20 or 25 degrees Celsius. So the enzymes in a plant have usually evolved so that their optimum is at a lower temperature. So notice one of the things we can actually do now that our techniques for studying biochemistry have improved, we could actually have a sample of an enzyme. And if we look at where that enzyme works best in terms of its temperature, we could make a pretty good educated guess about what type of living organism this enzyme might have come from. Now, what happens if the temperature is lower than the optimum or higher than the temperature? Well, above or below this temperature, either way, enzyme activity falls off. Now, what does that mean? The reaction that this enzyme catalyzes is going to slow down. Now, even though a high temperature, could make a reaction go faster, one big problem with a high fever is that if your temperature increases too much, remember, enzymes are proteins, so enzymes can actually be denatured, and then they don't work at all, period. And the problem is if these enzymes are for important metabolic reactions, a high fever could actually potentially kill a person. Now, a low fever can actually be useful. If you get a cold or a slight infection, one of the things that happens is you start running a fever, and a slight fever actually helps your immune system fight off an infection by a bacteria or a virus or a fungus or whatever it is. So we tend to think of fevers as being bad, but actually they're useful. If you take a class in anatomy, when you study the immune system, one of the things that you'll study is the fever response to an infection. Now, another way that we can affect enzymes is through some inhibitors. And inhibitors are chemicals that somehow interfere with enzyme activity. So let's look at competitive inhibitors first. So here's our enzyme, this gray blob. And notice that this enzyme has an active site. I don't know why they labeled everything else, but they didn't label that. So the active site is where this square is fitting it in up on top. We'll talk about the allosteric site in a moment. So this kind of peach colored thing is our competitive inhibitor. This green thing, you can kind of see if we took that peach out, we would have a half moon shape here. This green half circle, this is our substrate. So our substrate should fit into the active site, but a competitive inhibitor competes for the active site. And when it attaches, it blocks the active site. So since it blocks the active site, this means the substrate can't attach to the enzyme. So this reaction is going to slow down. Now, keep in mind, you have a lot of enzyme molecules, so it doesn't mean they'll all be blocked, but even if some of the enzyme molecules in a cell are blocked, the reaction will slow down. The other type of inhibitors we see are non-competitive inhibitors. Now, we see this kind of half square area down here that's called an allosteric site. Allosteric means different shape. And a non-competitive inhibitor can attach here. Notice when it attaches, it changes the shape of the active site. You can kind of tell up here the active site has a kind of a half moon shape for the substrate to fit into. When an non-competitive inhibitor attaches at the allosteric site, it changes the shape of the active site, and it changes it in such a way that the substrate can't attach. So again, this is going to slow down the reaction. A lot of pharmaceutical drugs that we use work as inhibitors. Sometimes they block bacteria from carrying out metabolic reactions, but sometimes they block your own cells from carrying out reactions. I've seen some commercials lately with Serena Williams talking about how she takes this medication for her migraine headaches. And after they talk about that, then they talk a little bit about the chemistry of how it works. They say, you know, what does this do? This blocks certain receptors on her cell membranes. So it interferes with those proteins functioning. Now, keeping in mind what we said, when we look at a metabolic pathway is going to be a whole series of related metabolic reactions. The 10,000 or so metabolic reactions that happen in a cell aren't unrelated. They're all similar to one another. Now, the way that they're related when we look at a pathway is the product of one reaction is used as the reactant for the next reaction. So let's think first about a linear metabolic pathway. And an example of one that we're going to study will be glycolysis. And it's called linear because it can be written in a relatively straight line. So here's a diagram of glycolysis. And our first reactant is glucose. And then down at the bottom, our last product is 2-pyruvic acid. And it shows us some little dots representing how many carbon atoms are in each molecule. So this is a linear reaction where we can basically go straight down from glucose to our pyruvic acid. Now let's look at a couple of the individual reactions. So remember, every arrow here in blue is an individual reactant. So glucose is the reactant. It's kind of at the beginning of this first blue arrow. The product that's at the end of this is something called glucose 6-phosphate. So glucose 6-phosphate has a phosphate group attached to the 6th carbon. Remember, glucose is C6H12O6. So the 6 tells us, you know, which carbon that phosphate group is attached to. Now, where did we get the phosphate? We got it from ATP. Glucose reacted with ATP Remember, ATP is adenosine triphosphate. It has 3 phosphate groups. ATP loses a phosphate group, so it's ADP at the end, adenosine diphosphate, and di means 2. So we've gone from 3 phosphate groups in ATP to only 2 in ADP. Now remember, the chemical bond connecting the 3rd phosphate to the 2nd phosphate has a lot of energy. So when we break that 3rd phosphate off, we use the energy that was holding it to the 2nd phosphate, and we use it to stick a phosphate, this kind of little yellow ball, onto the 6th carbon in glucose. So notice for our first reaction, glucose and ATP are our products. ADP and glucose 6-phosphate are, I'm sorry, glucose and ATP are our reactants. I think I said products. Glucose 6-phosphate is at the point of the arrow. ADP is at the point of the arrow. So these are products. So remember, reactants are at the beginning of an arrow, products are at the point. Now the second reaction, there are actually 10 reactions in glycolysis. The second reaction, glucose 6-phosphate is our reactant. So we just made it as a product. Now we're going to use it up as a reactant. And we're going to change glucose 6-phosphate to fructose 6-phosphate. And remember, glucose and fructose are isomers. They both have the chemical formula C6H12O6. The most obvious difference between them is in glucose, there's a double bonded oxygen on the 1st carbon in the chain. In fructose, the double bonded oxygen is on the 2nd carbon in the chain. Okay, so that's our second reaction. Our reactant is glucose 6-phosphate. Our product is fructose 6-phosphate. So glucose 6-phosphate was the product of the first reaction. It's the reactant for the second reaction. Now let's do one more. Fructose 6-phosphate, we just made it. Now we're going to use it as a reactant. So we're going to react fructose 6-phosphate with another molecule of ATP. So when we do that, we're going to pull the 3rd phosphate off of ATP again. And since there was a lot of energy holding that 3rd phosphate onto the molecule, we're going to use that energy to stick that 3rd phosphate onto fructose. So our product won't be fructose 6-phosphate, it will be fructose 1,6-diphosphate, or it's sometimes called fructose 1,6-bisphosphate. So di means 2. So where are the phosphate groups located? Well, there's one on the 1st carbon over here, and there's one on the 6th carbon over here. Then we're going to use fructose 1,6-diphosphate as a reactant. So we're going to keep doing this until we get all the way down to the bottom, till we get to our pyruvic acid. We're going to make a product, then we're going to use it as a reactant. So this is how our chemical reactions are related to each other in a metabolic pathway. We make a reactant, then we use it as, we make a product, and then we use it as a reactant in the next reaction. So we want to keep that in mind. Now let's look at a different type of metabolic pathway, something called a cyclic metabolic pathway. And a cyclic metabolic pathway can be written as a circle. And the key for why it's cyclic is the product of the very last reaction is the reactant of the first reaction. So let's look at an example. So we're going to take oxaloacetate, we're going to add acetyl. That makes citrate. Now remember our basic definition of a metabolic pathway. When we make a product, it gets used as the next reactant. We're going to take citrate and we're going to make ketoglutarate and carbon dioxide. Then we're going to take ketoglutarate and we're going to make succinate and carbon dioxide. Then we're going to take succinate and make fumarate. Then we're going to take fumarate and make malate. Then we're going to take malate and make oxaloacetate. Well, notice the oxaloacetate down here was the same chemical we started with. So let's look at a graphic representation of this. So here's our oxaloacetic acid, another name for oxaloacetate. Here's our acetyl. So here's citric acid or citrate. There's a few compounds in here I didn't put in for simplicity. But let's not focus on the chemical compounds. Notice the arrows represent chemical reactions, and you can see that ultimately we end up back at oxaloacetic acid. If all we did was kind of spin in a circle, there wouldn't be any point to a metabolic cycle. The benefit of a metabolic cycle is not that we end up with what we started with. The benefit is a metabolic cycle allows us to change some inputs into outputs. So let's look at our inputs. Our inputs are all written outside of the circle. So one of these is something called acetyl-CoA up here. Notice it's at the beginning of this kind of golden arrow. Another input is this NAD+, which is at the beginning of an arrow over here. Then there's an NAD+ and a CoA here. Then there's an ADP and an inorganic phosphate here. Then there's an FAD here. Then there's another NAD+ over here. So we have lots of inputs. What are our outputs? Well, let's think about this acetyl group. Acetyl has 2 carbons in it. So what's going to happen to these 2 carbons? They're going to come in as acetyl. They're going to leave as carbon dioxide. So these 2 carbon dioxides represent the 2 carbon atoms that came in as part of acetyl. NAD is going to pick up some hydrogens, and when it does, it's going to make NAD+. So here's an NAD+, here's another NAD+, here's another NAD+ over here. Notice coenzyme A is just going to leave as coenzyme A. Remember what I said, this is kind of a chauffeur. It picks up things with that YL ending at acyl groups and transfers them from one molecule to another. ADP and phosphates are input. ATP is our product. And FAD is our input here. FADH2 is our product. So the real value of the Krebs cycle, which is named after Hans Krebs, an old German biochemist who worked on the chemistry. But the real benefit of the Krebs cycle is not the cycle part, but the fact that when we go around the cycle, we convert these inputs into those outputs, because the cell is going to have some use for NADH and for ATP and for FADH2. Now, in our case, our cells don't have much use for carbon dioxide, so we exhale carbon dioxide. But instead of thinking about a plant cell, if an animal cell, if you thought about a plant cells use a lot of the carbon dioxide that they make. They need carbon dioxide for photosynthesis. So don't worry about all the details for now, but what you should worry about is the fact that you can follow the arrows for the reactions, and you can see that we end up with oxaloacetic acid, the same thing that we started with. And the other thing you want to think about is the fact that while we're going around this circle, we convert all these inputs into outputs. And the same thing with glycolysis back up here. Don't worry about all the details. Now we are going to come back to glycolysis and the Krebs cycle and look at the individual reactions in a little more detail later. But for now, you want to make sure you can distinguish between a linear metabolic pathway and a cyclic or a circular metabolic pathway. And then don't forget our discussion of enzymes earlier. Now, are there any questions about what we've talked about in terms of enzymes or metabolic pathways? Okay, I'm gonna stop screen sharing and